webinar. We're going to be talking about treatment and vaccines today. We are just waiting for our second panelist to join us, and as soon as he joins, we will start. We're going to give him 5 minutes. If he doesn't join us, we will just get started with Prof. Luis Enjuanes, that he's here, and he's joining from Spain. So just bear with us for 5 more minutes, and as soon as he's, here he is, perfect. Good afternoon, Willem. Thanks for joining us. I was just introducing the webinar, so I guess we can just get started. This is the second COVID webinar that the Association of Spanish Scientists in South Africa has organized, in this case with the Embassy of Spain in South Africa. So we are going to be talking about treatments and vaccines today. We're going to get started with a short video from our ambassador introducing the webinar, and then we'll go ahead with the presentation from Prof. Luis Enjuanes from Madrid, Spain, and follow up with a presentation by Willem Hanekom, and then we will have time for some questions and a discussion. If you have any questions, please go ahead and use the Q&A or raise your hand, and at the end I will give you some time. So thanks for that, and let me just share the screen so that you can all see this video. Oh God. Okay, sorry. I'm getting notifications that you guys cannot hear the video. I don't know how that happens, so I'm just gonna go ahead and do the introduction myself. Sorry about that. They say I can just enable sound. Okay, let me just. Okay, I'm gonna give it a second chance. Okay. Let me know if you can hear it.
Good afternoon, and welcome to the new series of 2 webinars organized by the Spanish Embassy in Pretoria and the Society of Spanish Scientists Researchers in South Africa. Those webinars highlighted the pandemic caused by COVID-19 and promote cooperation between South Africa and Spain. On the title Treatments and Vaccines, the 2 webinars are focused on the drugs and medicines that are currently being used or could be used in the future to recover from COVID-19, as well as on the production of those needed vaccines. In this first webinar, we present 2 of the most prestigious virologists of both countries, Professor Luis Enjuanes from Spain and Professor Willem Hanekom from South Africa, who I thank for accepting our invitation. Graduated in chemistry and doctor in virology, Professor Enjuanes is a research professor at the Higher Council of Scientific Research, head of the Laboratory of Replication, Virus-Host Interactions and Protection Coronavirus, and the National Biotechnology Center of Madrid. He is a member of the Spanish Royal Academy of Physics and Natural Science and of the American Academy of Microbiology. In 2007, he was recognized by the Spanish Society of Virology for his contribution to the development of virology in Spain. He is the author of more than 200 publications and 10 patents. Professor Hanekom is the director of the African Health Research Institute in KwaZulu-Natal. His contributions have included directing the South African TB vaccine initiative at the University of Cape Town and the TB vaccine program at the Bill Melinda Gates Foundation. He currently holds professorship positions at University College of London, University of Cape Town, and University of Washington. He is the previous chair of the South African Immunology Society and the Federation of African Immunology Societies, and is a member of multiple international advisory committees in tuberculosis, vaccinology, and transnational immunology. Thank you very much. I hope you enjoy.
Okay, so without any further ado, we'll give the floor to Prof. Luis Enjuanes so that he can go ahead with his presentation. And that's all from me. Go ahead, Prof. Luis, we can't hear you. Please, can you unmute?
Yeah, but you silence me, I think. Can you give me
sorry.
the voice?
Yeah, there you go. You can talk now.
Okay. Do you see the screen?
Not yet.
Not yet.
No.
I share the screen. I can restart again.
Yes, please.
Let me try. Share a screen again. Share. Do you see it now?
Yeah, we can hear it now. See it now.
Okay, then I expand the screen now. I hope I still can see the controls. Do you see it now?
Yes, we can see your mouse and we can see the screen.
Okay, thank you.
Thanks.
So let's get started. I will speak briefly on coronavirus pathogenesis and protection. I will review 3 viruses, 3 human coronaviruses. Coronaviruses, they belong to 5 different genus. Genus alpha, beta, delta, epsilon, and gamma. Genus alpha coronaviruses, they infect domestic animals and humans. Genus beta infects mice, also domestic animals and humans. Genus delta, wild animals and humans. Epsilon, bats. And genus gamma, chickens and birds in general. But the key point is that all these coronaviruses infecting all these animals, all of them, they come from bats. And the main point is that those bats are flying now over all continents, of course in Africa, Spain, and so on. So very likely they could re-emerge at any time, as it has been happening in the latest years. There are 7 known human coronaviruses. The first 4 in black are coronaviruses that appear in the 50s and 60s, and nowadays these coronaviruses are fully attenuated. They cause the winter common cold. But the other 3 at the bottom in red disappear in year 2002, 2012, and 2019, the SARS coronavirus 2 that is causing the actual epidemics. So these 3 viruses that I just mentioned appeared 2002, 12, and 19. They have infected in total around 8,000 people, 2,600, and the last one, coronavirus 2, 52 million people, and have caused 700, 800 deaths, 850, and more than 1 million people die. But the mortality is quite different. The SARS-1 was killing about 10% of the infected people. MERS coronavirus was infecting 35% of the infected people. And the present coronavirus is infecting only 2%. But because it has been distributed into 235 countries, it disseminates very efficiently, and it has been causing a pandemic, which is killing really many people. The first SARS coronavirus that appeared in year 2002 show up in the southeast of Asia, in the Guangdong province. And when that happened, WHO asked the authorities to chase any animal that was moving around and analyze whether it carried the virus. 3 different animals, civet cats, raccoons, and ferrets, were identified as infected by the virus. But the main one infected by SARS-1 were the civet cats that are considered in the Southeast China as a delicatessen and served in the restaurants. But when we study the evolution of the genome of this virus in civet cats and also in human beings, the virus was not stable, was evolving very fast, indicating that this animal, civet cat, was not the natural reservoir for the virus. In fact, it was demonstrated later that were the bats flying all over. This virus is causing severe and acute respiratory syndrome, pneumonia, high fever, and death. People associate this virus infection to a respiratory disease, but the virus grows even better in the enteric tract. In fact, when you follow a different days post-infection of a person, the presence of the virus in the nasopharyngeal aspirates related to the respiratory system, the amount of virus is related to the blue columns. And when you look to the feces of those patients, the virus titers are even higher, the red columns. And also the virus was present in the urine of the patients. Now, the second deadly human coronavirus that appeared in Saudi Arabia, in the Middle East, emerged in year 2012 and was extended to 27 countries. And there have been, in the last 17 years, many years, only 2,650 people infected, but this virus was causing the death of 1 out of every 3 persons infected. This is because the virus does not transmit well between human beings. The virus goes more efficiently between camels and men. In fact, the intermediate host, the one that is taking the virus from the bats, are the camels, and the camels pass the virus to humans efficiently. But we do not have too much contact with camels, so it does not spread out efficiently. The third virus, the present deadly SARS coronavirus 2, is coming also from bats, but we still don't know the intermediate host. It has been speculated whether it was the pangolins or the snakes, but that cannot be confirmed. What happens when this deadly human coronavirus, as 1 or 2 or MERS coronavirus, when infect an alveolus of the lung? Initially, the virus enters in the respiratory system and will infect the epithelial cells of the lung and also macrophages. Macrophages will be activated. They will be release a collection of factors. And some of those factors, they are chemoattractant for neutrophils that are circulating in the blood. Neutrophils are very peaceful cells that circulate, but if they are attracted to a tissue through those interleukins, immediately they go in, destroy virus, infected cells, but unfortunately also non-infected cells, and then the lung gets full with liquid, cannot interchange oxygen, and then the patient dies. With the actual virus, different negative properties. One is that it can cause asymptomatic infection for at least 14 days. That means the person gets infected, but he has no signs, fever, or any other thing. So the person will distribute the virus to many other people. They can induce a limited immune response and of the reduced length. They can re-emerge in patients that were already recovered. And the worst thing, probably in my opinion, is that those viruses they can be activated by a proteinase cleavage of the spike protein of the virus. What happens is that the spike protein of these coronaviruses that we represent by a bar, to be able to activate the virus to enter the cell, it needs to suffer two cleavage, two cuts in these two positions. But the important thing is the SARS coronavirus that appeared in year 2002 did not have an incorporation in this protein that allowed the cleavage by furin. And why is that so important? It's because furin is present in all tissues. Therefore, this cleavage site this is the spike protein located in this red dot, is already cleaved, and the protein is pre-activated such a way that the virus can enter in any tissue, and after second cleavage in S2 primer, it can infect cells. So before leaving an infected cell, these viruses, with this addition of the furin cleavage sites, they are already activated, and they have the power to infect basically any organ of our body, any tissue. And the big difference is that SARS coronavirus of year 2002, it was only able to infect respiratory tract and enteric tract, whereas the new virus can infect lungs, causing acute respiratory disease syndrome, lung edema and death, can infect the gut, kidney, brain, heart, veins, COVID-2, that is due to a malfunction of the blood circulation that causes a stroke, an ischemic ball, causes a smell loss, an infection of tongue, so it's causing also the loss of the taste, and in severe cases, it can infect also pancreas. Why is so bad this virus? It's because it drastically affects the immune response, particularly the innate immune response, which is acting immediately after infection. That reduces the response that appears in 3-4 hours, and even this virus during infection induces the production of autoantibodies against interferon. Interferon is a molecule that will defend us as fast as 3 hours after being infected. Also, a second huge problem is the production of a huge inflammatory cytokine production that stimulates too much our immune system due to the release of all these factors in red that do activate and attract T cells, natural killer cells, monocytes, macrophages, and neutrophils. Neutrophils are terrible because when they are activated, they immediately destroy all the tissues and cause pathogenicity. Another main problem is that the infection by the virus causes a dysfunction of SARS coronavirus elicited antibodies. There are many abnormalities in the induction of antibodies. And it has been observed when we analyze patients that they recover and compare them with patients that they die, in both cases, antibodies are produced. But in this case, people that recover, the antibodies are directed against the spike protein of the virus. Whereas in the cases of the people die, very few antibodies are directed against the spike. These are the neutralizing antibodies. That's the reason that these young people in general, they can be protected, whether the other people with antibodies against an internal virus protein, they die. And there are other abnormalities in the production of antibodies because they do not mature properly, because the germinal cells, where B cells that produce the antibodies, they not mature properly. Now, in this situation, what do we do? Which strategy we develop to protect against virus infection? There are 4 main approaches. One is the induction, restoring the natural immunity mediated in general by interferon and other molecules. The production of vaccines, I will focus today on that, but also the production of antivirals that interfere with virus replication are important, or the use of antibodies, either from plasma of people that were infected and induce an efficient immune response. This plasma can be administered to patients and save them. But also we can produce highly specific monoclonal antibodies that they are very efficient neutralizing the virus, and they can be produced in unlimited amounts. I will focus in vaccines, just describing very briefly what is this. There are different types of vaccines. Those that we call inactivated, that are based in an inactivated virus or a viral protein. They can be produced very fast, but they induce low mucosal immunity in the respiratory mucosas. That is the main area where we need them. And they do not induce lasting immunity. Another type is the use of live attenuated viruses. Viruses that by removing some genes on them, they do not cause infection, but they induce a good immune response. And because they mimic the natural route of infection, they are highly immunogenic and induce long-lasting memory. But there are biosafety concerns because although we attenuated these viruses, we cannot trust them because viruses evolve very rapidly and they can go back to virulent. The third category is the use of viral vectors, adenoviruses that have been attenuated, or vaccinia virus attenuated. They can be developed very fast. Their safety is very well documented, and they in general are very efficient. Most of the actual vaccines for SARS-CoV-2 are based on viral vectors. But in our lab, we like to work with even safer vaccines that we call RNA replicants. As we do not trust infectious attenuated viruses, we have developed from the genome of the viruses RNAs that can be self-replicate. They are self-amplifying, and they strong induce immune response, and they have a key property. This is a key word for me. They are propagation deficient. What does it mean? They cannot be propagated. They don't move from one cell to the other, and therefore they are highly safe. Inactivated virus will be based in the purification of a coronavirus like this, as we did in the past, and then chemically inactivated, it can be a vaccine. But as I said, we are looking for modern vaccines, and they are based in the understanding on the molecular basis of virulence. People think that a virus is virulent if it grows to high titers. This is not true. A virus is virulent if it carries in its genome the genes involved in virulence that are acting by neutralizing the immune response. So to be able to work on this system, we need 2 tools. One is to engineer an infectious clone. This is the genome of a coronavirus, and it's a genome that is an RNA. You cannot perform genetic engineering with RNA. You have to make a cDNA copy because that allows you to introduce cuts in the viral genome and remove different genes one by one until you identify why, by removing, for instance, gene E, as I will focus on, the virus is attenuated, and more importantly, it cannot be propagated. Therefore, it will be the base of a vaccine candidate. because anytime we attenuate a virus, we can immunize with this virus. It will produce no disease, but will induce an immune response. And we need an animal model, like a humanized mice, to test whether our viruses are virulent or not. Because these are deadly viruses, we need to be working every day in safety laboratories. And we have been deleting all viruses that are, all genes or the viruses that are non-essential. I focus in one now, gene E. E is the envelope gene. It's a gene encoding the envelope protein. In one infected cell, is shown in green, is highly abundant, the E protein. And look what the observation we made when we had a virulent virus and we deleted from him the E gene that we call delta E, and we inject this virus in humanized mice, and look what happens in their lungs. If we have deleted gene E, the lungs, they are normal, they can interchange oxygen, and the virus is attenuated now. If we do not delete the gene, you see there is infiltration in the lungs, and at 6 days post-infection, the air space is full with water. The animal cannot interchange oxygen, cannot breathe, and dies. So we got an attenuated virus. That was fantastic. So that was a vaccine candidate. We immunized mice then. And at different times post-infection, we saw whether this virus, 3 weeks later after infection, after the immune response was developed, whether it was resistant to a new infectious virus. In non-immunized mice, when we infect them with a lethal amount of virus, they will rapidly lose weight, and by day 4, survival will be 0. All the mice were died. But if we immunize those mice with a virus in which we deleted the gene, the weight is maintained and survival is 100%. That means 100% protection. This is the way we have generated our vaccine. But in fact, this is more complex. We have deleted 1 gene, E gene, or 2, or 4, or 5, in order to induce another safety measures. And the virus that we have generated is propagation deficient, because when we infect those animals and go back to the lungs and see how much virus is, if the virus is the wild type with no deletion, the virus can produce at day 3 post-infection more than 1 million of infectious particles. But if we have deleted 5 genes, as was the case here, as you can see in the orange columns, there is no virus growth. This is because the virus cannot move from one cell to the other. Therefore, it cannot go back to virulent. This is a key word. It cannot go back to virulent. It's a safe virus. We check that all these virus that we engineer, that now we should not call them virus because they are RNA replicant. Why don't call them now viruses? Because they cannot be transmitted. And when we infect with them and we check for the attenuation, we see that in fact all of them are attenuated, because when we infect mice, they don't lose weight, and when we check virulence, 100% of the mice survive. If the mice have not been, the virus has not been attenuated, they lose weight, and by day 8, survival is 0. So this is very important. And we immunize with these deleted viruses and study protection 3 weeks after immunization of those humanized mice. When 3 weeks after immunization, we challenge them with a lethal dose, 100,000 infectious particles. And we check what is happening. If the virus, the animals were not immunized, they rapidly lose weight, and by day 7, survival is 0. All mice are dead. But if they were immunized with a vaccine, they even gain weight, and survival is 100%. In fact, in both cases, when we have the wild-type virus, the virulent original virus, in the cytoplasm. They make viral particles. But even if we have deleted the gene, they also make virus-like particles, particles that are polymeric and therefore highly immunogenic. When we immunize with those particles, we get what we call an sterilizing immunity. That means that no virus can grow in the animals that we have immunized with our vaccine. Non-immunized mice, the virus black columns can grow at days 2, 4, and 6 post-infection to high titers, more than 1 million of viruses. But those that were immunized in orange columns, the virus doesn't grow. This is sterilizing immunity. The virus that tries to get into the immunized mice does not have the chance to produce infectious virus. And we have 2 delivery systems. In one, it's fully synthetic. We mix the RNA replicon with a polymer. We make nanoparticles. And by introducing those nanoparticles in the nose with a spray, they go to the lung and induce protection. Alternatively, we make those virus-like particles that you see in this picture by electron microscopy, and they can also provide protection. I think I will stop here. This is the people from the lab that has collaborated in these studies. So thank you very much for your attention, and I will wait to the next presentation for any questions that you can send to us. Thank you very much.
Thanks, Prof. Enjuanes. That was a really interesting presentation. As Enjuanes has said just now, we are going to go ahead with our second presentation by Prof. Willem Hanekom, and we will have all our questions at the end. So Willem, you can go ahead and share your screen.
Right. Thank you very much for this opportunity. In the era where, in the coronavirus era, we got most of our information from Twitter or from other social media, etc. So I just want to alert you to this incredible resource that therefore exists, and that is called the New York Times. So if you go to the New York Times to their website, you can actually look at various treatments and vaccines that are being tracked. And it's actually a fantastic resource to see exactly what's going on in terms of vaccinology for coronavirus right now. When Anna contacted me, I said, well, we're not doing any vaccine research ourselves or treatment research ourselves. We're doing other kinds of work in coronavirus. And so Anna said, okay, well, talk about that work. So unfortunately, there's going to be very little here about vaccines or treatments. We are at a group called Africa Health Research Institute, and we work in rural KwaZulu-Natal, which is near the Mozambican border. And our vision is optimal health of under-resourced populations. This is a picture of the kind of populations in which we work. And so this is where I am at the moment, right up in the Nkele area, for those that do not know South Africa. But we also have a campus in Durban, which is a much bigger city. And there we have pretty fancy labs, and when we, in the end, do transdisciplinary research. So we do research that's population-based, clinical-based, social science-based, basic science-based, to understand and to intervene in the health of South African communities. So until March 2020, our focus was very much on HIV, TB, and non-communicable diseases. And here you can see, for example, 2 kinds of results. One is about transmission corridors in the population that we work with that can be targeted by public health authorities for interventions.
Sorry, Prof. I'm getting messages that people cannot hear you properly. Apparently, there's a little bit of echo. I don't know if you can maybe get
Okay,
closer.
I'll try and get closer to the microphone.
Okay, thanks.
Is this better?
Can you try again?
Is this better if I speak like this?
Yes, it
Okay.
sounds better. Yeah, thank you.
Right next to the computer now. Okay, so the other example is just an example of a novel pathway that's been discovered in terms of for protection against tuberculosis by studying human lungs, excised human lungs, and Prozac actually works in synergizing with anti-TB drugs. So then we got to our coronavirus epidemic in South Africa. And this is what it looks like at the moment. We've had about 750,000 cases in our country. We had a massive epidemic. At one stage, we were the fifth highest in the world. But right now, we are at a tail, and hopefully we will not have the second wave that Spain and other places are having at the moment, but that may be optimistic. So to deal with this in KwaZulu-Natal, we actually formed a research consortium, where the groups that you can see on the right there, the PRESA, CRISP, ECF, AHRI, two universities, HSRC, MRC, and Think, who have never worked together in this space, started working together, all the principal investigators, and we had weekly meetings and to discuss various things in this space, or research topics in this space. And so for those that are on the call interested in, you may be interested that we're having a conference pretty soon that you can see the details of there. But we had various working groups working in different areas such as health systems, basic mechanisms, social science, viral sequencing, diagnostics, etc. Also clinical trials, one trial of experimental intervention, isoxazoline, and WHO treatment trial, and then 2 vaccine trials that are ongoing, but absolutely no results from any of them at the moment. So today, I thought I'd just show you results from 2 activities amongst these. So the first is work done at the Somkele campus up here in northern KwaZulu-Natal. And this is where we have got a health and demographic surveillance site led by Kobus Harps, who's the senior author on this manuscript that's going to be submitted. It's called Protective Behaviors and Secondary Harms from Non-Pharmaceutical Interventions During COVID-19. So basically, what Kobus and his colleagues did is they went to 3 such sites, which are demographic surveillance sites, where specific populations have been followed for years and years. One here, one in Mpumalanga, and one in Limpopo, and had nearly 14,000 participant contacts to find out what is happening surrounding the COVID epidemic and the lockdown to these populations. So clearly, most of the interviews with the populations were done by telephone, and this is obviously a picture from the pre-COVID era with no masks, but in our call center. And then if participants were identified that could have COVID, they were approached with our mobile services for interventions or for further management. Excuse me. So this is the epidemic curve that happened in the 3 provinces that I mentioned, and in black in South Africa. And at the bottom, you can see the visits, the contacts that were made with different groups of people over that period of lockdown. So we had the strictest lockdown, initially level 5, and now we are in level 1 lockdown. You can see the lockdowns at the bottom, and then you can see all the visits. So quite a few visits at all these 3 sites, all in different color represented here, to get the information. So the first thing was, how effective was the national messaging about COVID-19? And it was pretty effective. So these are the numbers of people that thought at the 3 sites, all in different colors, that they knew enough about COVID-19, and you can see it increased over time, but it was pretty high in the end, more than 80% at our site, for example. And these were the number of recipient of participants that said that they used face masks on a regular basis, and that was very impressive. And then these are the numbers of people that said over this time that they would be avoiding crowds. So pretty impressive also, so the messaging does seem to have worked. But Guy then went and looked in much greater detail at mixing that happens in these communities. So he did a study in 2019 to look at social interactions at our site here, and he could compare these results then. This is from a survey in June. He could compare these results with the results in 2019 and showed, for example, that there was much less contact, like between 15 and 29-year-olds and 65-year-olds, there was 44% less contact than there was 1 year ago. So there was actually less contact, but there were still concerns in that the people at highest risk, such as people over 65, still had a lot of contact with people at lower ages. The impact of mental health surprised us a little bit. I think the communities were extraordinarily resilient, and these are two markers of anxiety and depression, or standardized markers, tools that were used, and the rates were pretty low. When we looked at healthcare access, there were some concerns. So, for example, it looks like overall, around about 50% over the period, 50% of participants over the period could not access their medications. And this was a pretty important thing. And when we broke it down, it looked like child health care, after the lockdown was introduced in the red, was the one that was most affected by the lockdown. So why are we doing this kind of research? This kind of research is fed directly back to the government and impacts the interventions that happens in these communities and even nationally. So the conclusions were that these populations are extraordinarily useful for generating quick results. The messaging was effective by the government. There was concerns about ongoing social mixing, especially with older people. There was relative resilience in mental health, and there were concerns about impact on healthcare access. Okay, and now sort of a turn more to the Durban side. Here we did this study, which was a study to look at what does HIV do in terms of COVID outcomes. And in this study really focused on immunological outcomes, although we will really show some clinical outcomes too. So the core question here was, is the immune response different in persons who are HIV infected or not infected? And for those that do not know, in KwaZulu-Natal, more than 1 out of 3 persons are HIV infected in this province that we live in. Okay, so this is the demographics or the characteristics of the populations that were enrolled in this immunology study by Alex Alex Siegel and his colleagues. There were 124 participants. Their ages were pretty similar, in their 40s. Most were females, as we see often in clinical trials and studies that we do. There were sort of equal amounts of comorbidities in the 2 groups of HIV negative, HIV positive. And then at the bottom, you can see how disease was characterized in terms of asymptomatic, mild, or requiring oxygen. So in this study, there were no participants that actually needed ventilation or further intervention. So before I go on, I have to say that when we also looked at symptoms between the two groups, which was not really the aim of this study, and there was really very little difference in symptomatology, although this is a small sample size to look at symptomatology. The first question that was asked is what happens to shedding of this virus in people living with HIV? So do they shed the virus for a longer time than people who do not have HIV? And here you can see in blue uninfected persons and in red infected persons in various categories that you can see, either suppressed or, virally suppressed or viremic. And you can see there doesn't seem to be too much of a difference. Perhaps there may be some people that are HIV infected in which the shedding may go on for a longer period. The next thing that Alex looked at was T-cell depletion. And so we know from quite expansive literature right now that T cells are depleted during this viral infection, as is during many other viral infections. And so here you could also see, according to disease severity, HIV infected in blue, uninfected in blue, the more severe the disease was, the more depleted the T cells were, and the same sort of pattern was also seen in HIV infected people. So that was no surprise. And then, but what Alex could do was look longitudinally at how these cells expanded after they were diagnosed and followed up. And what one can see is basically in HIV-infected persons, especially on the left in the red, that there was a pretty dramatic expansion, at least in the peripheral blood, of T cells after during recovery from this viral infection. Alex also decided to look at various markers that pertain to various immune processes in this cohort. So, for example, CXCR3, which is a homing marker, PD1, which is obviously a marker associated with exhaustion, HLA-DR, and CD38, which is associated with activation. So basically, the only marker that showed some patterns amongst these was really CXCR3. And this is what we saw, is that the more severe the disease is, the lower the expression of this marker was. And then, and it was pretty impressive that if you were SARS-CoV-2 positive, if your nasal swab PCR, that the CXCR3 levels were significantly lower, the expression levels of CD8 cells, and this is a completely novel finding. And this is what happens over time. It looks, especially in HIV-infected people, that the expression does return to baseline after, well, what we presume was the baseline after the disease is over. They also looked at antibody-secreting B cells. These cells are CD19 positive, and secreting cells are CD27 and CD38 positive. And here the patterns were not that dramatic. These are what we saw according to disease severity. And then over time, this is now in the entire group, which is in green, there was a decrease in the expression, in the number of these cells. So early data to work on, but the data that in the real world, in a relatively resource-constrained environment, show what the effect is of HIV infection on the immune response. So perhaps the conclusions could be that viral shedding may be prolonged in HIV infection, that there's an impressive CD8 T cell expansion over time in HIV-positive persons, and that the decrease in the CXCR3 expression happens decrease happens when HIV-infected persons are virus positive. So that is all I have. So thank you very much. Not much vaccinology or treatments, but as Anna asked, I've given you a flavor of the kind of work that's going on in KwaZulu-Natal in this area. Thank you.
Thanks so much, Willem. So we have some time now for questions. I've asked everyone to please use the Q&A or the chat to put your questions so that we can go ahead and answer them. So I'm going to start with Shishia posted a question. I think it's for you, Prof. She asked regarding the social contact study from Guy Hartlin's work. Is that contact between teens and elders mostly between household or family members?
Anna, is it possible to open this up to other people also, so that Guy can answer directly?
If you can, if we can ask Guy to just raise his hand, I can allow you to talk. If he's in the attendees, and he can just raise his hand, I can go ahead and do that. Is he?
I'm not sure. I cannot see the participants here.
I can't see him in the list.
Anyway, either Guy or anyone else can answer, but I'm not entirely sure. I think that's mostly in the household, the interactions, but to be honest, I don't know the detail of that.
Okay. Shishia, we can find that out for you and just let you know. So we have a raised hand by Rousseau, so I'm just going to ask you to unmute so you can go ahead and ask your question. I don't know if... Well, nothing is happening. Okay, so I'm just going to go ahead and read the questions from the Q&A. So someone is asking, if we're living in a TB endemic country and assuming we have all been in contact with TB and carry TB bacilli, I don't know how you pronounce that. Is it possible that the ongoing innate monocytic response to TB is protecting us from coronavirus?
Who asked that brilliant question? So I really like that question. So they're talking about the concept of innate immune training, which is a pretty modern concept which has been described, for example, after BCG vaccination, where it appears that that innate cells such as monocytes are being trained, although they're not part of the adaptive immune response. These innate cells are trained to better respond to pathogens if they encounter them in the future. So, for example, BCG vaccination in newborns may protect against other diseases, viral diseases perhaps, especially in resource-poor environments. So I think that may actually be what is happening. I think there's a hypothesis that exposure to many pathogens would allow a lot of immune training, to use simplistic language, and therefore be protective to some extent. It remains a hypothesis because there are areas like South Africa, in India, for example, where there's also massive exposure to pathogens all the time, including tuberculosis. And it doesn't seem as if immune training has saved India from this massive epidemic. But in South Africa, for some reason, we had a huge epidemic, but we are on a plateau now, and perhaps that may be contributing to that.
If I can add something, do you hear me?
Yeah, yes,
okay.
Go ahead.
In Spain, there is a team doing research on the TB vaccine for a long time. And in fact, they got permit now to see whether people that is administered the TB vaccine, whether that will provide any protection to SARS-CoV-2 patients. And it has been approved for clinical experiments phase 1, 2, and 3, because the preliminary work was that in fact the TB vaccine, as Professor Willem Hanekom was indicating, they pre-stimulate the immune system and they provide a non-specific protection that alleviates pretty much the infection by human coronavirus, SARS-CoV-2. So it's making progress now and it's been tested in human beings in different countries, including the United States and of course Spain and other countries.
Yeah, so that's probably Carlos Martin and his
yes, correct.
Yeah. So
correct.
yeah, and the very simple BCG is also being tested in 5 major studies all over the world, and to see if it's going to make a difference. And apparently, we will have the results of the big BCG studies within a month's time. So very exciting.
Okay, we're going to continue. So Nseba Keleni, I hope I pronounced that at least close to how it's pronounced, sorry about that, is asking Prof. Enjuanes, when do you foresee your vaccine will be in clinical trial stage?
Well, this is a question that will come better, I will say on Monday, okay, next Monday. Why is that? We have basically developed the vaccine candidate, but now we are expanding the production to small amounts, relatively small amounts. We have performed one experiment to produce the virus, to do the experiment in humanized mice. We already got the humanized mice susceptible to the virus. And tonight, we are getting the results on the titers of the amount of virus that we have generated. So if the titer is good enough, in 2 weeks, we will start the evaluation of our vaccine candidate in the mouse model. If it will work, then early next year, we will go to macaques, preclinical experiment. And if that works, then soon we will go to human trials phase 1, 2, 3. Altogether, that means that our vaccine will not be ready until the end of next year, but we know from our previous experiments that the vaccine was inducing sterilizing immunity. Very few vaccines that are now in the market, even those in phase clinical trials, they induce sterilizing immunity. I only know about one of them, so we don't worry about that. What we need also is that it will not cause side effects. This is very important. It will be safe for sure, because it's, as I described during my presentation, is an RNA that is propagation deficient, so the virus will not have the chance to go back to a virulent version. And for us, this is essential. Otherwise, we will not be able to recommend our vaccine candidate based on this RNA replicon to be used in human beings.
Thanks for that. So our next question is from Dr. Diego, and he's asking, it seems that anti-vaxxers are increasing by the day. How would you, once any of the vaccines become available, promote vaccination worldwide?
How all, if I understood properly, these vaccines will be available? Well, my personal opinion, having worked for more than 40 years in virology and 35 on coronaviruses, I think that we, the regulatory agencies, they have to make sure that the vaccines are really safe. I mean, it's acceptable if the vaccine provides good protection to have minor side effects that you can treat with an aspirin or a paracetamol or something like that. The vaccines that now have been developed, they induce minor effects like a little bit of fever, a little bit of headache, or muscular problems, and they can be treated with a paracetamol. So that's okay in view of the high benefits. But to make sure that this is the case, if you evaluate a vaccine candidate for 2 months and you see no problem, then all you can guarantee is that the vaccine is causing no side effects in the next 2 months. But this, in my opinion, is not enough. I think vaccines should be evaluated at least for 6 months in human beings. If after 4 or 6 months you see no major side effect, which is negative, then I think you can approve the vaccine. Therefore, since the most advanced vaccines are already being tested now in human beings, I think it would be safe to wait until the end of the first trimester of next year and to recommend the administration in general, because otherwise all you can guarantee is this vaccine is safe for the next 2 months, but this is not enough in my opinion. We should expect at least 4, I would say better 6.
Okay, thanks. Willem, do you have anything to add to that?
Yes, I think vaccine hesitancy is now a complete field like pulmonology or cardiology, because it's such a huge problem that there are people that have literally religious beliefs. And the basis of this may not be rational, and rational arguments don't apply often to people who are vaccine hesitant. So it's a very, very difficult thing to approach. And I think when the authorities that have to deal with this need to access true vaccine hesitancy experts and how to approach this. I'm afraid in what I've encountered, I lived in Seattle for a while, and in Seattle, it has the highest unvaccinated childhood populations in the United States, in the most liberal area of the United States. I have encountered some of this, and I feel that nothing is rational. So I think it's a very complex issue and something that really has to be addressed.
Okay, thanks. David van der Meer is asking, is there any reasonable indication that the use of BCG revaccination to prevent COVID in healthy subjects?
We don't have the results yet, but we'll have results within a month or 2.
Yeah.
Shishia is asking if there are also some suggestions that partial cross-reactivity to circulating human coronaviruses can provide some immunity against SARS-CoV-2, but there is not very much info on pre-pandemic human coronavirus epidemiology in this part of the world that she knows of. Do you guys have any comment on that?
Well, I can comment that according to different publications, 3-4 sounds from Germany, from one of my colleagues, they claim that, in fact, if we have been infected in the past by one of the 4 attenuated coronaviruses that emerged in the 50s and the 60s, the T-cell response can provide some protection. In fact, they have proposed that some of the people that they have been infected and they become asymptomatic for a long time, this is because the protection from the T-cell responses. Hopefully that will be true, and as I said, that has been claimed for at least 4 different laboratories, one from Europe, from Berlin, and Christian Drosten, a very well-known coronavirologist, He has been attending on our meetings for many years, and 2 or 3 papers from United States. So then in general, I think there's an agreement on this area, and that will be good because basically every adult person, at least in the Western world, but I assume will be the same in Africa, has been infected more than once with these attenuated coronaviruses, because all they cause is a winter common cold, very gentle, that you don't have to stop going to work because it's all you need is to take a paracetamol, and if the T cells from those viruses, stimulated by those viruses, provide protection, that is great. That will be very helpful. And it looks like this is the case.
Okay, thanks. So we're going to get one last question, and then we'll try to adjourn. So we have a question for Prof. Hanekom. Do you foresee AHRI playing a role in vaccine development?
Yes, definitely. We may even be, as you know, we are setting up a clinical trials unit right now. It's in a very careful and staged approach. We hope to start our first clinical trials in July next year. That's our timeline. And it may well be a COVID vaccine that would be the first that would be tested. Don't know, but that may be.
Okay.
May I ask one very last question?
Go ahead. Yeah, I was going to open for you.
Yeah, the question is, I follow this issue very closely. I always have been very interested. As a virologist for many years, I have observed that viruses, when they jump from animals to human beings, they are highly virulent, but they do not disseminate efficiently. And that with time, they fix mutations in their genome, and that helps them to be more efficient in the dissemination. But fortunately enough, this is also associated to the attenuation of the virus. In fact, that happened with apparently with the older 4 coronaviruses. All of them are now attenuated. And that happened also with SARS-1, when that emerged in year 2002. Initially, it didn't grow efficiently, but then it was learning very fast to disseminate properly. But at the end of the 6-8 months of the epidemic, the virus was already attenuated. Therefore, I was hoping that with this virus, it will be happening the same thing. And I made the mistake of, based on my previous knowledge, to make this prediction, the hope of this prediction. Have you observed in Africa that the virus has evolved and you have identified a certain degree of improvement in the dissemination, as we have seen in Europe, for instance, but a reduced virulence? I can tell you that when I check with Spanish doctors that are very well located in very important hospitals, the answer is no. They don't see any attenuation for the moment. But that depends on the density of the population and how easily the virus can transmit for the selection. Have you observed any attenuation at all, or this is not the case?
I'm not sure I'm qualified to answer that question, but I will take it to a person who in our consortium who is doing these sequencing study, viral sequencing studies, and doing phylogenies and tracking what's happening to the virus. But I'm not sure of that answer. I don't think so. I haven't heard of that at all.
Okay. This is bad because probably is not being attenuated. Thank you.
Yeah, I was just going to make a comment that I don't think we've had enough of a big wave to start seeing that changes this quick. Maybe by next year, middle next year, we might start seeing something like that. But then again, it will be interesting to see the sequencing data and figuring out if that's the case.
Your numbers of infected people are excellent. They cannot be compared with our numbers. In Spain, the average number of infected people in the second wave is higher than 500 for each 100,000 people. Oh. So these are very, very high numbers because obviously the density of the population here. This is happening also in Italy and not that big numbers in France, in Germany, but in the second wave is becoming really terrible in the European countries. Not only in Spain, Italy, France, Germany, United Kingdom, it's terrible.
I'm actually coming up with more questions, but I'm gonna have to cut it here because I know you guys are like busy right now, especially in this time. So we've been asked if it's possible to share your presentations. So if we can get those, okay. So we will just share all in our channel. We're gonna post the recording of this video in our YouTube channel, and we'll share their presentations as well in our social media. And thanks everyone for joining. We're gonna have a second session of a COVID seminar on the 24th of November with another speaker from Spain and another speaker from South Africa. So just keep a lookout for our next webinar information. And thanks everyone for joining. Thanks for both of you for such wonderful presentations. And that's all from me. Thanks.
Okay, thank you very much. Bye, bye, bye.